Good evening, everyone. Welcome to 𝐸𝐴𝐵 Academy live session today. I'm just waiting for the viewers to join. Today, I'm going to talk about general topic. What type of career opportunities do you have once you have a medical degree? There's a lot of confusion for the junior doctors. So what to do once they finish MBBS? And they all come to me, most of them, they come to me during breaks, and then they will ask me, is it the right profession? That's number one question. People try to get some answers from me constantly during my coffee breaks. So that's why I thought I can do this session today as a live session. And we have a special guest as well, Dr. Vijay Kumar, and I will introduce once I complete my presentation. So what type of career opportunities one have once you've done the medical degree? Once you got a medical degree, what is the next step? Number one, you could be just a consultant in your own specialty, or you can progress to become a medical director if you work very hard. You can become a clinical director when you become expert in your field, and you can become a professor if you're well into academia. So if you publish a lot of papers and if you supervise lots of students for 3 years, you may get a job as a professor in a university. You can be a lecturer, and you can be a tutor if you can't progress to become a professor. And obviously, if you don't want to teach students and if you just wanted to practice your own specialty, you can become a consultant. If there is no opportunity to become a consultant, you can be an associate specialist within that specialty. So these are the clinical jobs. So obviously, if once you finish a medical degree, these are the career opportunities open for you. Door is open to become a medical director, clinical director, professor, lecturer, tutor, consultant, and associate specialist. But some doctors, they don't want to practice clinical jobs. Maybe they are fed up with the stress in the work environment, or maybe they are fed up with workload. I'm not sure. So I interview lots of junior doctors, and they're most likely, no, I'm not interested to become a doctor anymore. I don't want to practice like a doctor. So these kind of negative thoughts people do get, and I want to change my profession. So that kind of attitude is there in some of the juniors, and a lot of them, like 20% of them, almost like 1 in 5, fail to recognize that they work hard to become a medical doctor in the first place. So this is why I wanted to streamline your thoughts today with this presentation. So yes, these are the highest clinical jobs you can get it: medical director, clinical director, professor, lecturer, tutor, consultant, and associate specialist. But there are even better jobs if you don't want to be a clinical doctor. So this is why it is important. That is why today I'm going to touch base. Medical degree is not a waste. You worked hard to attain your MBBS degree, which is not practically possible for most of the students. So what are the non-clinical jobs? If you don't want to be a hospital doctor, there are non-clinical jobs. You can be a best histopathologist, and you can be a microbiologist, famous microbiologist. You can get a Nobel Prize, and you can be a radiologist sitting in the beach somewhere in Australia and can report images. You don't need to be in the hospital, so you can work online, off-site. So, and also you can be a forensic expert. So you can give a expert witness, and you can become a research associate for a big companies, medical companies, or vaccine companies. You can be an academic professor. So you don't need to actually do a clinical work, but you can still be a professor as a non-clinical doctor. So this is why there are some non-clinical jobs which will also be as equal as the clinical jobs, like histopathologist, microbiologist, radiologist, forensic expert, research associates, and academic professor. Well, if you can't even get to, these kind of non-clinical jobs, there are, other non-hospital jobs. But first of all, I would like to touch base what are the hospital jobs which you can get as a post-MBBS doctor. You can get a foundation training job once you finish your master MBBS degree. you can do a foundation training jobs and you can apply for master's degree, without master's degree, you can, once you finish your foundation training jobs, you can go into core training and do membership exams and progress to specialist training. And if you finish your specialist training, you can become a consultant. And if you're not in a training, you're trained abroad and you want to do some jobs for money, you can do non-training service jobs. And ultimate aim is to become a consultant in the hospital jobs. But if you don't prefer hospital jobs, that's where today I'm going to explore the avenues. So what are the non-hospital jobs? There are people who became CEOs, Chief Executive Officer of a trust, for example, Aarogyasri's trust, like some health programs, some health schemes. You can become a Chief Executive Officer and you can become a Chief Operating Officer of a trust. So you can become a data scientist and you can become a director of a company. Today I'm going to introduce a director of a company who runs the pharmacovigilance, and you'll have a first-hand experience what it feels like. And you can also become medical director in a pharmaceutical company elsewhere, like in states. Lots of my friends have become medical directors in states. And ultimately, if you don't want to practice in the hospital, there is another area, primary healthcare, where you can become a GP, general practitioner. So general practitioner, you can make equal money and more money, and lifestyle will be very good. If you don't want to practice a hospital job, you can be a non-hospital doctor in a GP practice. But obviously, if you have a lot of skills, you can become a dean of a university or a dean of a big firm, basically, which has got lots of universities under its radar. So, if you don't want to practice anything clinical, you can still become a medical superintendent in the hospitals, which is also very highly rewarding job. So there are so many non-hospital jobs, and these are, today I'm going to introduce a person who has become a director of a pharmacovigilance company at the end of this presentation. So why do we need to change profession? This is really very, very great challenging things. Number one is, you came all the way to become a medical doctor and you finished your MBBS. Now you want to change your profession, which doesn't make sense in real world, because lots of people are struggling to get an MBBS degree, and lots of them have appeared entrance exam and they couldn't get through, so they're struggling in their lives. And now you got an MBBS degree in hand, and you want to change your profession, desperately change profession. You don't want to be a hospital doctor. You don't want to be a non-hospital doctor. You don't want to be related to anything related to medicine. So there are some other professions, like you can become a medical statistician, you can become a scientist, you can become entrepreneur, you can become an IAS officer. There are lots of people who became IAS officers in India, and you can be a scientific advisor to the state, and you can be a chief medical officer to the state, and you can be a chancellor, vice chancellor. You can become a health minister if you're aspiring to go higher and higher. So, but our idea is, these are all extreme situations, like becoming a medical statistician, becoming a scientist, becoming an entrepreneur, becoming an IAS officer, or scientific advisor to the state, or chief medical officer, or a chancellor to the university, or a health minister. These are high-end spectrum. Not everyone can achieve that. That's why. So I'm going to introduce a person who is established himself as a pharmacovigilance director of a company, Dr. Vijay Kumar. So non-hospital jobs. I was explaining that you can be a entrepreneur or you can be a pharmacovigilance director. So as you have first-hand experience as a director of a company in pharmacovigilance, so I would like the viewers to listen about you a little bit. So Dr. Vijay Kumar, would you be able to tell what you do for your living, Dr. Vijay Kumar, as a post-MBBS as a doctor?
Hello, hi everyone. Sorry for the technical thing. I need to back in. So, as introduced by my friend and colleague, Dr. Malik, I'm a qualified physician. I got my qualification in 2002, 2003. So and to answer the question of non-clinical, non-practicing medical jobs. So I have been in pharmacovigilance for the past 15, 16 years. Started my pharmacovigilance career in 2007. So after my medicine, I was practicing. I was pursuing my medical experience and aiming for getting a postgraduate degree, etc. But pharmacovigilance came across, and I've been in pharmacovigilance since 2007. It's a medical job, but non-practicing. So you don't see the patient on a daily basis, like face to face, but you interact with patients. You still use your medical knowledge. You still use your training experience. You still use your medical books to answer the question. So. Pharmacovigilance is a very interesting field. Medical professionals particularly has a very good opportunity to become a pharmacovigilance physician. You can go to any levels. You can be the chief scientific officer. You can be a medical director. CS, we call it a chief scientific officer. Um, uh, in a nutshell, pharmacovigilance has very good opportunities for doctors who really want to go into a practicing field. So I know doctors who do both, like they practice as well as also do pharmacovigilance in their spare time. So it's a very good career opportunity. Yes, Malik. Yeah. Any other questions?
Can you hear me, Dr. Vijay Kumar? Can you hear me?
Yeah. I can hear you now.
Okay, so I would like to ask a few questions, Dr. Vijay Kumar. How do you feel yourself in a non-clinical job without practicing in a hospital? How do you feel about changing your career to completely different pathways? How do you feel yourself? How do you see yourself, as a non-clinical doctor?
I wouldn't call me as a non-clinical. I would call me as a non-practicing, like not practicing. So it's like
Sorry, please
like,
correct. Okay.
like any other job. So let's say you finish your medicine, you finish your post-graduation, and you choose to go into a academic field. So you teach students. So you're not treating patients. You're not talking to patients. Does it mean that you're unhappy? To me, if you're interested in any field very relevant for your field, I I don't see any difference whether you practice or non-practice. You still use your knowledge. You still use your experience and expertise, and you still contribute to the patient's good and patient safety. So to me, personally speaking, it is a very satisfying job, and I don't regret or I don't feel anything different than a practicing physician. As long as you like your profession, you should be happy in it.
That's great. So, are you missing something from hospital? Like we practice day to day life, we touch patients, we have bit of satisfaction and gratitude, and we come home and say, "Oh, we cured some patients." So, do you miss anything of that kind? Being a non-clinical doctor, sorry, non-hospital practicing doctor.
I just miss the interaction with the patients. That's all. I mean, I don't get a chance to see patients face to face like regular practicing clinician. But I talk to patients on a daily basis. They call the company. They call my company to clarify a lot of things about how to use a medicine. What are the risks? Using pregnancy, using breastfeeding, etc. So I have daily conversations with the patients relatives, just like you see in a GP surgery or in a hospital setup. Yeah, I miss the interaction, the face-to-face interaction. That's the only thing I miss. Otherwise, it's all the same for me. I'm still as I.
Okay. Can I ask you a bit more question about what could be the financial impact or implications about changing your profession altogether into a pharmacovigilance industry? So how do you feel about how it impacted your life financially?
To me, if you like something, if you like something and you enjoy doing something, I think you'll find ways to explore and open more doors where you can make equal or more money than a regular practicing physician. So, to give an example, I mean, in the Western world, in UK, Europe, North America, pharmacovigilance experts like myself, like experts can be any life sciences background person, like a pharmacist or anything like that. But for doctors, they make equal or even more in some situations, than what a regular practicing clinician makes.
That's good to know, Dr. Vijay. So you can make more money than a consultant. Do you agree with that?
Absolutely. So if you consider my example, right in front of your eyes, so I run a limited company. It's a one-man company. It's not like a typical corporate where you have an office, tables, and a lot of people. Actually, it's a one-man company. So I'm the sole director and CEO of Drug Safety Consultancy Limited, which is established in 2016. So if you take the turnover, the last the last, the latest annual turnover was more than 185K in British pounds. I don't have any employees. I don't have any apart from the regular maintenance and stuff. So I don't pay anybody else. So that's the whole turnover of the company. So you can make up your own mind based on that.
Okay. There's another question I would like to ask. What is your long-term goal? You established as a director of a pharmacovigilance company. So is there any long-term goal? Like, would you like to start your own company, pharmaceutical company, or do you have any other things in your mind? So what could be the end point for this career?
As I was mentioning and probably disconnected, so I would like to establish a global CRO. So I mean, see, it's up to you where you want to go. You want to settle in a particular once you reach a particular peak in your career, or there are always doors. You need to open them. You need to explore them. There are opportunities. So if you have time and money, all the right project plan, right contacts, you can establish a global CRO, without actually having to set up any office or anything like that. So you can save money there. So virtual CRO, offering services globally rather than locally. Yeah, that would be
That's
the.
very good. That's very good. So, last but not the least, one more question I would like to ask. What do you suggest for upcoming doctors who have passed their MBBS and who doesn't like the hospital work, and who are like in the first 1 or 2 years itself, they don't want to be a clinical or non-clinical doctor. So, what would you recommend? For the upcoming doctors, would you recommend this kind of profession, like a pharmacovigilance industry and other professions like IAS and those kind of non-clinical jobs?
I mean, I can't speak on behalf of everybody. Like I think it's a very difficult. Very difficult process. It all depends upon the candidate. It all depends upon the individual in which field that particular, as you want to go to academic teaching, drug safety, pharmacovigilance. So it depends. And if you really, I can only speak for pharmacovigilance. If anyone wants to become become a professional in pharmacovigilance, be having a medical qualification, which is a very advantageous, which puts you already on top of the group of individuals with a pharmacy background or etc. So if you have a medical qualification, you're already ahead of the game. So I can only say in pharmacovigilance, there are huge number of opportunities, just like any other medical, the opportunities in medical field will never run out. Like, would there be a day when pharmaceutical companies stop manufacturing products? I don't believe so. It's not possible. So for humankind, it is not possible. So companies will continue making products, continue bringing up new therapeutic areas, new therapeutic lines, which nobody else have discovered. So that will continue. So as long as that continues, pharmacovigilance, which is dealing with drug safety and safety of the patients while using medications, will never stop. So it all depends upon the individual. So pharmacovigilance, if you really decide to become a pharmacovigilance professional with a medical qualification, the door is out there. You just need to open it and then explore.
Thank you very much for your inspirational speech today, Dr. Vijay Kumar. Thank you for joining us, and we'll definitely invite you once again in future for other live sessions with us. So, if anybody would like to ask the question, we are happy to take the questions. And for next another 5 minutes, we have time to before we go to special topic about bias, risk of bias assessment in the meta-analysis. Before we go to next topic, so we would like to take some questions. So Dr. Vijay Kumar is here for another 5-10 minutes at least, so that he can answer any questions. If viewers want to ask questions, please ask questions at this stage. Thank you very much for your inspirational speech today, Dr. Vijay Kumar.
You're welcome. Thank you.
Any questions, please? Viewers can ask questions while we have 4 minutes to start next presentation on bias and blinding. So please. So if you don't have any further questions, we're going to proceed to next special topic today, which is bias, risk of bias assessment in the meta-analysis for the included studies. So what is bias? Bias is, as we are speaking last week, we in a meta-analysis concepts, one of the concept is quality assessment of the studies, which we covered elaborately last week. And another topic I would like to cover today is risk of bias assessment before we head into heterogeneity testing in meta-analysis. So what is bias? Bias is nothing but a systematic error encountered when you're conducting a study or when you're doing a trial. So it's a systematic error. What's the difference between a bias and a random error? So let's say, for example, if you have a scale which has wrongly displayed units, and you measured with the same scale all the measurements using the same scale in your study, and then you come to know at the end that the scale itself is wrong. That means you committed error across the whole study systematically. That is what is called a systematic error or a bias. Why is it so important that if there's a bias, what happens? If there's a bias, you can get false positive results or a false negative results. You can encounter both type 1 or type 2 errors. So you deviate from the truth, you deviate from the results, and your inferences will not reflect what is there in the primary objectives, and the inferences doesn't match with each other. The primary objectives deviate from the results. So as a consequence, there is either underestimation or overestimation. So you may not be able to truly quantify the intervention effect. So that is why risk of bias in a meta-analysis needs to be addressed before you do heterogeneity testing, because it leads to methodological heterogeneity and then statistical heterogeneity. So obviously a bias can be quantified as well. We'll talk about that, how to quantify a bias, and what is impression and what is bias. So as we all know, bias is a systematic error committed from the time of conducting study to the reporting the results and even publishing the results. So whereas imprecision is nothing but you actually use a scale and you don't know how to use that scale appropriately, and you actually measured wrong measurements. As simple as that. Imprecision is different from systematic error, which is bias. So the scale itself is wrong. That is where the bias is. So if your scale itself is wrong, that you committed a systematic error, that is where is the bias. Imprecision is you don't know how to use the scale. That's why you committed error. That is what imprecision is. So either way, if there is a risk of bias, it affects the quality of the included studies and quality of your results. So what are the 5 major domains of the bias? So there are 5 major domains of the bias. One is a selection bias. You wrongly selected patients into that study. Or performance bias. So you don't know how to recruit the patients, either recruitment or while you're conducting the study, there are issues. Either it could be dropouts, it could be anything. So if it's a performance issue, like you haven't delivered the tests or you haven't given the treatment adequately, so that is where the performance issue is, performance bias. Attrition, some of them can drop out in the midway, and then you don't notice them until the end. Then there can be attrition bias. Detection bias, your test maybe fail to recognize the positive result and you get false positives or false negatives, then there is a detection bias. And finally, the reporting bias. You don't know how to interpret the results. Either it could be units wrong or it could be wrong measurements. Either way, so there is a reporting error. The actual results encountered is different from actual results reported, or you might be hiding some results. So it is called reporting bias. So these are the 5 domains of the bias which normally you encounter if there is no adequate blinding. So selection bias, performance bias, attrition bias, detection bias, and reporting bias. So it's important that is why Cochrane collaboration tools for assessing risk of bias elaborately tells that it is important you do a sequence generation, allocation concealment, where you put it in a covered envelope so that the person who is conducting the trial, the participants doesn't know, participants are blinded, person who is conducting the trial is blinded, person who is evaluating the trial is blinded, statistician is blinded, even sometimes the person who is interpreting the results is also blinded. So blinding of participants, personal and outcome assessors is so important. You need to blind all from participants to assessors, you need to blind them. So when you encounter there is a incomplete outcomes, then you need to report them. So the data of the incomplete outcome should be reported and should be found out very well in advance. And it is bad concept that you report selectively. That is amounts to bias again, reporting bias. So there can be other sources of bias like funding issues, or even attrition, dropouts of the patients, or suddenly abrupt stopping of the trial. So there are so many types of biases that can affect the trial's results. So to summarize, in a meta-analysis, you can classify the studies into low-risk studies, unclear risk of bias, and high-risk studies. So once there is a high risk of bias in those studies, then you may have to either omit those studies or do meta-analysis separately with high-risk studies and separately with low-risk studies. So I have given various definition what is meant by low risk and unclear risk and high risk in this table. So obviously the low risk is low risk for all the 5 domains we talked about. So 5 domains of risks of bias. If there is 1 or 2 domains affected, it is low risk. If it's more than 2 domains, then it is intermediate risk. If it is more than 3 domains, it's high risk. So 1 domain, I would take it low risk. 2 domains, I would take it unclear risk of bias. 3 domains, it is high risk. As simple as that. So you can assess the risk of bias when there is when you encounter bias, what happens? So when there is a clinical heterogeneity and when there's a bias, then you'll obviously get a heterogeneity when you do a heterogeneity testing and a meta-analysis. So then you can't progress to do a meta-analysis, do you? So in that case, you have to do a meta-regression or do a subgroup analysis. So that you try to see if homogeneous groups, is there any difference between homogeneous groups related to interventions like males and females. So we can even quantify the risk of bias. So as I said, high risk bias studies into one group, low risk bias into second group, and you can calculate odds ratio for high risk studies. You can calculate odds ratio for low risk studies. So the formula for quantifying the risk of bias is a ratio of odds ratio of the interventions odds ratio in studies with high risk or unclear bias divided by intervention of odds ratio in studies with a low risk of bias. So on the if you have a high risk studies as a numerator, if you divide the odds ratio of the high risk studies versus that of low risk studies, that will quantify the risk of ratio of or quantify the risk of bias by doing a ratio of odds ratio. If it's more than 1, that means that the odds ratio, if it's more than 1, there is a definitely bias in that meta-analysis or meta-regression. So it's nothing but ratio of odds ratio, where you get an odds ratio for the high-risk studies, an odds ratio for the low-risk studies. You divide the odds ratio of the high-risk studies with that of low-risk studies, and that is how you quantify the risk of bias. If you encounter risk of bias more than one, that means that there is a risk of there is a bias in the included studies. Therefore, you cannot progress to do a meta-analysis with high-risk studies. As simple as that. If you do that, you will encounter false positive results or false negative results. That is why it is important to blind the participants in this study. So that's why when you are doing a meta-analysis, it's important you assess the studies for whether there is a blinding, participants, healthcare providers, assessors, data analysts, and manuscript writers even. So whether there's a blinding is followed as per Cochrane tools, we need to assess these studies before you do a meta-analysis with those studies. So that is why it is important. So key concepts is whether there's a blinding in selection, detection, performance, reporting. So those 5 domains which I spoke earlier. So we need to assess the risk of bias in relation to those, and they're all linked to blinding in one form or the other. You will know whether who is blinded, who is not blinded if you read an article which is included for meta-analysis carefully. That's why it's important also you assess the risk of bias when you're assessing the outcomes in each article which is published, whether they're subjective or objective, or, how they concluded their results, so you can assess. That's why it is important that Cochrane tools of bias in risk assessment of bias should be followed in all the quality assessment of the included studies when you want to perform a meta-analysis. So that is why I would encourage all the people to use those 5 domains which I stressed about in assessing the risk of bias in the included studies. If you have any study which is qualifying, which is encountering 3 domains of the bias, that means it's a high risk study, and you may have to omit the study in the meta-analysis, or do a meta-analysis with high risk studies separately and report them separately, and low risk studies separately and report them separately. So this is all in order to prevent heterogeneity, statistical heterogeneity. If there's a methodological heterogeneity, you will definitely encounter statistical heterogeneity. As simple as that. That's why risk of bias assessment is a major part of the quality assessment and prior to heterogeneity testing in a meta-analysis. Why is it risk of bias is so important? Because it affects your results. It affects your validity of your meta-analysis results. So that is why it is important you need to identify the potential threat to validity of your results of meta-analysis. So there can be so many factors that can be affecting or validate that are going to cause potential threat to validation of your meta-analysis or meta-regression results. It could be design-specific risk of bias. It could be early stopping of the trial, or it could be baseline imbalance of the participants in the included participants in the trials, or it could be randomization issues. It could be differential diagnostic activity, or maybe they would have reported just interim results and they would have not waited till the final results. So it could be they altogether they would have deviated completely from the protocol and would have done the study differently. So you need to see whether they stick to protocol from beginning to the end. So is there any contamination? Is there any null bias? What is null bias? Is nothing but interventions being insufficiently well delivered or over widely inclusion. If you have a wider inclusion criteria, then you will encounter a null bias. That's why you should have a definite inclusion criteria. You set inclusion criteria when you start to do your meta-analysis. What is your strict inclusion exclusion criteria should be defined prior to do a meta-analysis. So that's why most of the time, you set some inclusion criteria and then you don't follow them. When you're doing meta-analysis, then you actually encountering null bias. or they might have reported instance to instrument results in those studies, so you need to be careful. And also sometimes selective reporting is quite common when they do subgroup analysis, and sometimes it could be fraud. Some results can be maybe some companies would try to portray very good result. It could be a fraud, or it could have been inappropriately influenced by the funders. So some clinical trials can have this kind of institutional manipulation, and inappropriate influence from the funders. So we need to be careful. That's why these are all the potential threat for validity of your results. So that is why when you're selecting studies for meta-analysis, you need to look for all these things, like, are there any potential threats for validity? So if there are potential threats for validity, you will encounter bias. And then obviously when you encounter bias, there is a methodological heterogeneity that will lead to statistical heterogeneity, and full stop, you can't do a meta-analysis. And so you need to progress to do a meta-regression instead of meta-analysis. Therefore, risk of bias assessment is mandatory, especially against those 5 domains at least, if not all the potential threats to validity. I've enlisted quite a lot of potential threats to validity apart from 5 domains which I explained in the beginning of this presentation. I hope I made it very clear that it's important at least you test risk of bias in the included studies for at least for those 5 domains, if not all the threats, so that you don't get heterogeneity. If you do get heterogeneity, then obviously you end up doing a meta-regression rather than meta-analysis. I hope I made it clear today. And if you guys have any questions, please feel free to ask at this stage. So the next topic we're going to talk in a couple of weeks is about heterogeneity testing. So in 𝐸𝐴𝐴𝑝 Academy, we teach about meta-analysis and meta-regression concepts. So for Masters and PhD students, we are here to help, and please subscribe to our online lecture modules. We will do a live session and demos how to do risk of bias assessment, and also how to do heterogeneity testing, and how to construct forest plots. So in a couple of weeks, we're going to do a live session how to do heterogeneity testing and what are the various types of heterogeneity. So once you're familiarized with risk of bias assessment, we will proceed to heterogeneity testing. So please watch this video again and get familiarized before attending the next session. So I hope you guys have enjoyed my talk today, and also the talk from our special guest today about other career opportunities for doctors. So I hope you enjoyed my video today and live session. Please subscribe to www.𝐸𝐴𝐴𝑝 Academy and register with 𝐸𝐴𝐴𝑝 Academy for more updates, and also subscribe for online courses. There are free courses available on the 𝐸𝐴𝐴𝑝 Academy, and also some paid courses for Masters and PhD students. Please visit 𝐸𝐴𝐴𝑝 Academy. And if you have any queries, please drop me an email at info@𝐸𝐴𝐴𝑝.academy. So we are now doing live sessions related to meta-analysis series. So as a part of this, we have done some live sessions on quality assessment of the studies and risk of bias assessment today, and the next one is the heterogeneity testing in a couple of weeks' time. And I'm waiting for viewers to ask me questions. We got 5 minutes time, so please feel free to ask any questions if you would like to. I hope you all enjoyed my session today. In a couple of weeks, we're going to talk about heterogeneity testing. Please drop me an email if you have any questions on risk of bias assessment at info@𝐸𝐴𝐴𝑝.academy. Thank you, and I hope you enjoyed my session today. Thank you, all of you.